Hi, I'm Andrea Munsell.
And I'm Christiana Georgi.
And we're going to be talking about the HIV reproductive cycle.
Okay, so here we have the HIV molecule. And inside the molecule is the RNA. And we also have the enzyme reverse transcriptase. And that is enclosed by a protein coat called the capsid, which is also enclosed by the viral envelope. And surrounding the viral envelope are the glycoproteins. So this is the capsid, and these are the glycoproteins. From there, the HIV molecule goes into the host cell, and it releases the RNA and the enzyme reverse transcriptase. And from here, the RNA attaches itself to the enzyme reverse transcriptase because that's how RNA forms to DNA. So from here, the RNA becomes RNA-DNA hybrid. Which then forms into just DNA. And from there, it goes into the nucleus.
Okay, we are now in the nucleus. And these two strands we have right here are the chromosomal DNA within the host cell. And what happens is the DNA it embeds itself onto the chromosomal DNA, like so. And this is then called a provirus, meaning it will never leave the DNA host cell nucleus at all. So from there, transcription occurs and mRNA is formed. mRNA moves out of the nucleus and two jobs are done with it. It becomes, here's the mRNA. It is translated into glycoproteins, which are what these are. These are glycoproteins. And the glycoproteins come towards the cell wall and start to form. Those are them. And then we have an RNA genome right here that makes itself ready for other generations of the virus. So we have our capsid being formed, and then inside goes, the capsid is formed from the glycoproteins, and the other proteins formed after translation, which happens right here. And the RNA is inside of it. And then from there, we have it move towards the cell wall, and again, we have the DNA inside with our capsid. And then our viral envelope starts to form, right along with our glycoproteins. The host cell does not die. Instead, the virus is pushed out of the host cell, and a new virus is formed and it attacks other host cells, and those host cells can be white blood cells in humans, for instance. So to pull it all together, we have our RNA inside, the new RNA that we had. We have our RNA transcriptase. We have our capsid made from the mRNA. We have our glycoproteins. And we have our viral envelope. And from here, it all starts all over again. And that is the HIV reproductive cycle.